Hi friends, I Robinson. Welcome to our YouTube channel Start Science Robinson. Let's get into topic. Spleen. Today we are going to discuss about the spleen anatomy and functions. First we'll see the structure of our spleen, and before going further just we'll see about measurement of the spleen. So the length is around 5 inch, that is around 12 to 13 centimeters. And next, if you see about the breadth, it will be around 3 inches, that is around 7 to 8 centimeters. And the thickness of the spleen is around 1 inch, that is 2.5 centimeter. So this is about the height and the breadth and thickness. Now we'll see about the weight of the spleen. The weight of the spleen is around 200 grams, and it is been related to 9th to 11th rib. So we'll see about the relations of our spleen. So superiorly it is diaphragm, and inferiorly it is around left colic flexure of large intestine. Anteriorly fundus of the stomach, and medially it is pancreas and left kidneys. So spleen has 2 ends, 3 borders and 2 surfaces. So ends are anterior end is expanded and posterior end is rounded, and 3 borders, superior border is notched near the anterior end, and inferior border is rounded, and intermediate border is also rounded. So 2 surfaces, diaphragmatic surface is convex and smooth, and visceral surface is concave and irregular. And we'll see about the main functions of spleen one by one. So spleen, it plays an important role in immune response. At the same time, it is wedged and tetrahedral in shape, and this organ lying between fundus of stomach and the diaphragm. And spleen highly vascular and dark purple in color, and the main function is phagocytosis. There will be old abnormal erythrocytes which will be destroyed in spleen, and breakdown products bilirubin and iron are transported to the liver via splenic and portal vein. So next, storage of blood. This is around 350 ml of blood which will be used in any hemorrhages occurs, and it is also used in immune response. Erythropoiesis, spleen and liver are the important sites of fetal blood cell production. Outer covering of a spleen, it is covered with capsule as is shown in our diagram. So now I am showing you the partitions in cross sections of our spleen. So that is called as a tubercle. So after seeing that, we'll see about the blood supply to the spleen and what will be the structure of arterioles. So now I'm showing you about the arterioles which has been inserted into the spleen. So now we are getting into the spleen. So now I'm showing that in a diagram. So you just remember, so the name of the artery is splenic artery, and once it is been inserted into the hilum of the spleen, when it is introduced into the spleen, so then it is called as a central arterioles. So the branches which all been called as a central arterioles. So you can just remember and surrounds of your blind end of your central arterioles. There will be no directly connecting to the vein. At the end of the arterioles, the follicles are present, and the surrounding of the follicles again and the arterioles, the periarteriole are present, that is periarteriole. So lymphatic sheath, and surrounding of this periarteriole lymphatic sheath, there will be again marginal zone. So the marginal zone also will be surrounded. So as I'm showing in dots in our diagram, so it is a marginal zone, and these three, so follicle, periarteriole, lymphatic sheath and marginal zone, this is called as a white pulp, it is around 25% in our spleen. In our diagram now we are seeing about the splenic vein, so I'll just explain and what will happen in our splenic vein. So right now I'm writing it as a splenic vein, and now as it enters into the hilum of our spleen, as it enters into the spleen, it is called as a collecting vein. So after collecting vein, I'm writing right now it is venous sinus, where the orifices which is being collected from the cord and from the slit, where the normal orifices will be flowed from the cord to venous sinus and collecting vein and to splenic vein. Now we are seeing about the pulp. It is around 25%, that is follicles, at the same time periarterial, lymphatic sheath and marginal zone. Now we are seeing about the splenic vein collecting venous sinus and cord. It is around 75%, that is called as a red pulp. So now I am showing you the slits between the endothelial cells of venous sinuses, so where the old abnormal RBCs will not pass into the slit, so where the phagocytosis will occur. And I'm showing you in diagram about the white pulp, that is 25%. We'll see what are the functions in that white pulp, that is around 25%. I will just draw in a broader way. So already you know what are the things which is been follicles, arterioles and lymphatic sheath, at the same time marginal zone, which will made up of 25%, that is white pulp. So we can see if it is a blood which is flowing from the arterioles, central arterioles, and when it enters into the end of the arteriole, we have a dendritic cell plus antigen, and in periarterial lymphatic sheath, where you can find a T cells present. So in periarterial lymphatic sheath we can see T cells, and in marginal zone we can find a macrophages, so that is present in a marginal zone. So after seeing this macrophages, and you just see about the follicles where B cells are present. So in follicles B cells are present. So now we'll see when the antigen enters how immune response will occur in detail. We'll see here. So for example, if we see about the dendritic cells plus antigen, and which will activate our T cells which is been in periarterial lymphatic sheath, and which will activate in follicles, that is B cells, and the B cells will activate plasma cell. So the plasma cell which will produce antibody, so that is IgM and IgG, so which will fight against antigen. So that is the thing which will happen here in this 25% of white pulp in spleen. Viruses or any other organism which enter into the blood end of our spleen, where the follicles are present, where the B cells will activate and plasma cells will be activated, at the same time antibodies will be produced. Virus enters still, then the macrophages will activate and T cells, and T cells will activate B cells, and B cells will activate plasma, and again the antibodies will be released in our arteries. Then we are seeing about the splenic vein, that is collecting vein, inner part of our spleen, the collecting vein, and where the slits are present, where the macrophages are being situated. And now I'm showing you how the macrophages will phagocytosis the abnormal erythrocytes are destroyed in the spleen, and the breakdown products bilirubin and iron are transported to the liver via splenic or portal vein. So the RBCs which is been normal will enter into the slit and enter into the collecting vein, and it will enter into the splenic vein. So this is the process where the filtration, the abnormal RBCs will be filtered in our spleen through slits which the abnormal RBCs will not enter. So the phagocytosis will be done by the macrophages in a red pulp. So the RBCs which is been normal, which will enter into the collecting vein and into the splenic vein. So this is how the white pulp and red pulp in our spleen will work together to get rid of the infections in our body. So 1/3 of platelets will be stored in our spleen, so it will be sent in emergency situations when the crisis occurs. At the same time, 350 ml of blood will be stored in our spleen, so it will also will be used if any hemorrhages occurs. So this is about the spleen anatomy and functions. So you have to remember about the hilum, where the artery, veins and nerves will enter into the spleen. So that is the major portion you have to remember about. So there will be 25% of white pulp and 25% of red pulp. Thank you friends. It's Start Science Robinson. Thanks for watching.